Thank you everyone for being here today listening to this talk. I'm Jennifer Giron, a postdoctoral researcher at the Smith Lab of Insect Biodiversity at Purdue University, where I manage the Coleoptera Anatomy Ontology project. I'm also the acting collections manager at the Invertebrate Zoology collection at the Museum of Texas Tech University. Today, I will talk to you about the Coleoptera Anatomy Ontology and how it can be used for making morphological information more broadly usable and connected to multiple avenues of research. Since the time of Linnaeus, we taxonomists have described well over 420,000 species of beetles. Those descriptions usually highlight diagnostic features including size, shape, color, sculpturing, and the always important antennal shape. At first, descriptions were very brief, but over time, more detail has been added as more diversity keeps being discovered, along with the creation of multiple words to refer to multiple structures within a beetle in different groups. Nowadays, most descriptions incorporate dedicated sections to describe features of the head, the thorax, and the abdomen, detailing meaningful phenotypic variations that make species unique and distinguishable from other similar species. One thing that all those 420,000 species descriptions have in common, and at least double if we think of all the species that have been synonymized over time, is that all of them have been produced in what is called natural language, which is essentially the way in which we humans communicate with each other. And that has worked well so far. After all, we have collectively described well over 420,000 species of beetles in over 250 years. As we specialize and become familiar with the morphology and terminology that previous authors have used for describing species in our groups of interest, several issues become apparent with existing natural language descriptions, all of which stem from the fact that each of us has their own way to go about describing a species. Some of us would start by picking a model description to follow and adjust. Then we choose certain terminology that for describing the characters that we considered important, using certain words for describing shapes and colors, following a sequence that makes sense to us, which usually goes with the way we look at our specimens under the scope. And we all make decisions about the amount of detail that we put in our descriptions, which, in my experience, varies a lot depending on how many descriptions you have written and how much variation you have observed in your group, so that more experienced taxonomists tend to focus on key characters that they know that vary, whereas a beginner taxonomist would probably spend way too long describing absolutely everything that they can look at with the hopes of not missing anything that might be of importance. One key element here, and this is more a recommendation for taxonomists in general, but more so for experienced taxonomists, is that figures that are well labeled are extremely helpful, especially when learning the morphology. We keep hearing that taxonomists are at risk of extinction, but if we don't make it easier for others to understand what we mean in our descriptions, that might be a reason why beginners may feel inadequate as they can't quite grasp your descriptions, especially when reading through description in a foreign language that might interpret wording differently. So if for some reason you cannot add labels to your figures, at least point at a well-labeled reference in your methods if that exists. Going back to the point where we each have a particular style for writing our natural language species descriptions. One problem with them, besides comparability, is that there is no way for machines to be able to capture the wealth of information that is encoded in those descriptions. But why would we want to have machines be able to do that? Basically, it would allow us to optimize resources by enabling the extraction of information from the literature and by automating at least part of time-consuming tasks, including species descriptions and character coding. Okay, and how are we supposed to produce machine-readable and processable descriptions? Well, I'm here today to present to you the Coleoptera anatomy ontology, which we consider is the key to unlock the knowledge that has been encoded in taxonomic descriptions for centuries. An ontology in the context of informatics is a formal representation of organized knowledge so that this structured knowledge can be interpreted by machines. We already have a general understanding of how to organize what we know about the anatomy of beetles, which is actually reflected in the way that we structure our taxonomic descriptions. What we need now is a tool that can incorporate terms, their definitions, synonyms as presented by different authors, and graphic depictions of those structures across taxonomic groups. Before getting into details about the construction of the Coleoptera anatomy ontology, let me give you a little bit of background. When I was a master's student learning about weevil morphology, I came across the Hymenoptera anatomy ontology, which includes nearly every single term that has been used in Hymenopteran descriptions over time, and in which every term has a definition, with most terms illustrated, so it serves as an illustrated glossary for the order. A few years later, I met Aaron Smith, who was working with the Hymenoptera ontology team to produce the ontology for beetles. At that point, I got involved with the Coleoptera ontology as a side project. At the time, the ontology was being built in a specialized platform, which is no longer maintained, that was called MX, which was the same platform that the Hymenoptera anatomy ontology team used for the construction of that one. When I finished my PhD, Aaron decided to hire me as a postdoctoral researcher to develop the Coleoptera anatomy ontology. And to be completely honest, if there had been a formal advertisement for this position listing all the skills needed and which I have developed on the job, at that time, I would have probably not even applied for it. And this is just to say that as biologists, we gain a bunch of skills that can be further developed and polished over time. For example, in my case, project management and the interaction with a broad range of collaborators outside of biology. Since MX was not a viable option to use, we needed to find a way to create an ontology from scratch, but that followed community standards. For biomedical ontologies, the organization that sets and checks ontologies, that sets and checks those standards, is the OVO Foundry. For an ontology to be accepted by the OVO Foundry, it needs to follow certain rules, including interoperability with other accepted ontologies, which include Uberon, which is a multi-species anatomy ontology at higher levels, PATO, which is the phenotype and trait ontology, RO, which is the relation ontology, and more. The way to establish this interoperability among ontologies is by reusing terms that are available in existing ontologies and that have compatible definitions by importing those terms into the newly created ontology. Then, the ontology has to pass through a series of reasoning tests before going through an evaluation process. To ensure that new ontologies conform to the principles of the OVO Foundry, they created the Ontology Development Kit, which automates several ontology building tasks and quality control processes, allowing for collaborative editing using GitHub. Given that there was no backbone insect ontology that we could reuse to nest the beetle ontology, and since the Hymenoptera anatomy ontology is not fully compatible with beetles, we took on the task of building the ontology for the anatomy of the insect skeletal-muscular system, or AISM, which is a general ontology for insects that aims to serve as a controlled vocabulary where every single term has a logical definition that can be interpreted by machines and a natural language definition that humans can easily read. It also serves as the backbone for order-specific ontologies. It has been built using the Ontology Development Kit, and it's edited using a program called Protege. Most of the development of the AISM was done in collaboration with Istvan Miko and Luis Gonzalez, with comments and feedback by several other colleagues, including people in the ontologies world. One of the things that we wanted to achieve by creating this general ontology for insects was to be able to provide both natural and logical definitions for every single anatomical term. To accomplish this, we came up with a model for describing insect anatomical structures, which incorporates structural properties, so indicating if a particular structure is a sclerite or conjunctiva, or if its surface is flat, protruded, or depressed. The model also incorporates positional relationships in the form of locators, such as indicating the tagma where the structure is found and specifying if it is dorsal or ventral within that tagma, plus pointing other structures that are connected to the structure of interest. By incorporating the continuity and the connectedness of the cuticular system into our definitions, we can make each definition unique within the ontology and also unambiguous, which are requirements for ontologies to be internally consistent, and also help determine the most appropriate term to use for referring to a particular structure. As an example, here we have a tibia, which in the AISM is defined as part of an insect leg, an appendage segment, and a ring sclerite, which is bilaterally paired. It is adjacent to the distal margin of the femur, where it is encircled by femorotibial conjunctiva, and adjacent to the proximal margin of the tarsus, where it encircles tibiotarsal conjunctiva. And here you can see how that looks in the ontology editor, where we have our term nested within a hierarchy with statements for its logical definition down here, which have been translated into a natural language definition that has been added as an annotation to our term. One important thing here, and the reason why we can link this ontology to other ontologies, is that each term gets a unique identifier, which allows to find terms with their definitions and annotations on the internet once the ontology has both been accepted by the OVO Foundry, and ingested by a portal called OntoB. Here you can see how the term tibia is connected by different relationships with other terms. For example, these green links are showing that the tibiotarsal conjunctiva here on the right is encircled by the tibia, and the tibia, which is here in the center, is encircled by the femorotarsal conjunctiva. You can also, which is here at the top, you can also see that there are terms derived from tibia linked at the bottom in blue, which correspond to the pro, meso, and meta tibia. Here is a screen capture of the AISM in OntoB, where you can see that the main annotations for the ontology are here at the top, including a link to the GitHub repository and a brief description. You can also use the search feature that is there at the top in keywords to find terms within the ontology, and the content of a search will show you where in the hierarchy your term is nested, along with its definition and basic annotations. The AISM is also available for exploring at BioPortal, where besides navigating through terms, users can also visualize how terms are linked to other terms. We already used terms and definitions from the AISM in a paper looking at characters of the female terminalia of a tribe of darkling beetles. As you can see, there are some terms and definitions that are missing the AISM identifier, which is mostly due to those terms being taxon specific. So with the backbone provided by the AISM, we went ahead and created the Coleoptera anatomy ontology, again, using the ontology development kit, which means that it is already available for collaborative editing on GitHub. In its current version, released in November, the ontology contains over 600 terms, with about 40 of them being Coleoptera specific terms. Those beetle specific terms and their definitions were extracted from the morphology section of the first volume of Australian beetles, which I think most of us would agree that is the most comprehensive and up-to-date standard for general beetle morphology. You can see here for each beetle specific term that we have Coleoptera ontology identifiers. Also, for each term, we added the definition or general description of the term as found in the literature, along with the appropriate reference. Then we have the logical definition expressed as semantic statements here at the bottom, and a translated version to a natural language definition, which is annotated with an identifier for the person who constructed this definition, as well as the date when this definition was created. In the case of terms that come originally from the AISM, we can add synonyms when there are different terms in beetles to refer to a structure with the same definition. We can set that beetle-specific term as the preferred label in the Coleoptera ontology. And that is basically at the point where we are at right now, adding more beetle-specific terms from different sources, trying our best to match together terms that have the same definition. One of the big challenges here is that, as you might have noticed, anatomical terms are rarely defined in the literature. Most structures are described, of course, but not technically defined. This fact represents one of the barriers to make beetle morphology easier to process for beginners. That is why this ontology serves as a glossary to find definitions for beetle-specific anatomical terms. At this point, we are calling the community to help us curate this resource. There are two main ways in which people can get involved. One, by becoming part of the editing team by directly downloading the whole repository from GitHub and editing the ontology file using Protege. And if there is enough interest, we can possibly have a small workshop to demonstrate how to do this. Although we also have a manual that explains the whole process step by step. If it sounds too much like a hassle to learn how to deal with GitHub and Protege, another way to contribute is to go through the ontology, which is available in BioPortal. There, you don't have to download anything, and you can directly search through all the information associated with the terms and check if the definition is correct, or if you would like anything added to it, if we are missing key references for certain terms, if terms that are used in your particular group of interest are missing and need to be incorporated, and then contact us using either issues on GitHub or simply by email, specifying the changes that you consider that need to be made. Another way to contribute that perhaps involves less work is by sharing with us links to openly available annotated images representing morphological features of your group of interest. This way, we can use the links to annotate terms in the ontology, crediting the authors of the images. Okay, so now that we have a working version of the beetle ontology, how do we use it? The most basic way to use this ontology right now is by adding an ontology table to your species descriptions. We will incorporate this into upcoming descriptions and taxonomic revisions that are in the works by members of the lab. Some journals, especially PennSoft journals, already offer semantically enhanced marked up versions of the papers that they publish. So far, they link to taxonomic names in the GBIF taxonomic backbone, which you can see here highlighted in green. And they also link to publications using their BOIs, which you can see here highlighted in blue. So it would probably not be too difficult to also mark up anatomical terms using the unique identifiers linking to the ontology. The next stage is to start producing descriptions using simple semantic statements. One application for this, which is currently under development by Sergei Tarasov, is called Phenoscript. And what it does is to bring ontology terms in the form of a snippet, which is essentially a text file that you can set up in a plain text editor. Then in a new document, you can start typing your terms and relationships, and the editor will show you the options that are included in the ontology that you are using from your snippet. Each statement will be composed by a subject, a predicate or relationship, and an object. An advantage of this system is that because of the way that statements are constructed, using correct grammar becomes irrelevant at the time of generating the statements, which can be advantageous for non-native English speakers. Since these are individual-based descriptions, you can include information such as taxon name, unique identifier for the specimen, measurements, and more. These statements can be exported as OWL files, so ontology files, to be able to run reasoners and make inferences across taxa. Also, they can be used for semi-automatically translations into natural language descriptions. The idea is that these elements will be eventually incorporated into TaxonWorks. For those not familiar with this platform, it is an online set of tools that allow to manage and integrate taxonomic data, ranging from nomenclature and its associated literature, geographic information from specimens, so it can be used as a collections management system, and also information about morphology that can be in the form of storing images and creating character matrices. Another nice thing about TaxonWorks is that it allows for collaborative work. And the piece of good news, especially for Webull researchers, is that they already have a taxonomic database with over 26,000 Webull names waiting to be used in taxonomic projects. They are hosting informative sessions next week, including demonstrations, and everyone is invited to attend, so check it out. So whenever Phenoscript is incorporated into TaxonWorks, the terms from the ontology can be incorporated into all aspects of a project in TaxonWorks, including the generation of species descriptions from character matrices, for example. Okay, and what about what I mentioned earlier regarding the extraction of morphological information from the literature? Well, there are already applications that are able to process text and recognize sections within that text, like the software that is used by Plazi for extracting taxonomic treatments from publications. In a similar way, it is not very difficult to think of creating or adjusting applications that can recognize terms from text and associate them with terms in an ontology. So essentially, what we need is to get together with programmers or software developers to create or expand tools to make morphological data easier to use, perhaps in projects involving machine learning or artificial intelligence, for example, aiming to create large-scale morphological datasets. One quick tip here is that there is an NSF funding opening for infrastructure capacity for biological research to develop software for data mining and integration. So there are potential opportunities for funding research using ontologies in a biological context. From here on, there are multiple possibilities for incorporating structured anatomical information into several avenues of research. For example, anatomical data can be analyzed in the context of homology hypothesis testing, which requires other interacting ontologies, including taxonomic ontologies and the evidence and conclusion ontology. There can also be applications in evo-devo studies where transformation at the cuticular level can be specifically tracked during development. And it is also possible to link specific genes to specific morphological structures using the gene ontology and the protein ontology as well. So essentially, the Coleoptera anatomy ontology opens a broad range of research possibilities by providing a bridge across fields and across granularity levels. I will just finish here with a brief summary of what I talked about today. The Coleoptera anatomy ontology exists and can be used in several ways, including education, When we look at it as a glossary, it is also a tool for linking and extracting morphological knowledge from the literature, and is also a bridge to link morphology with other fields of knowledge. It is up to us as a community of coleopterists to make use of this tool. We can all help refine it to improve it, and we should all start adopting its use in our work when dealing with morphological data. We also need to start forming partnerships with software developers who can help building the tools that our community needs to make our life easier by using computational power to be able to create and analyze large-scale phenotypic data. By adopting the use of the coleoptera anatomy ontology in biodiversity research, we can contribute to increase links within the biodiversity knowledge graph. By generating information about phenotypic traits that can link to taxa and taxonomic names, linking those traits in publications and linking them to specimens by using their unique identifiers, we can also link to images depicting those traits and to people as authors of particular definitions of a given term. We could also link traits to phylogenies and even sequences when we get to involve the gene ontology, for example. This project has been funded by the US National Science Foundation, and I would like to thank all my collaborators for being there every step of the way to make this project a success. I'll be happy to answer any questions that you might have. Thank you.